So when a mummy and daddy love each other very much, they have a special hug. No, I'm just kidding. This isn't going to be a story about that. But I am going to tell you about something that happens soon after the sperm and egg meet. So about 7 weeks after conception in humans, our gonads go through a process called sex determination. This is the process where the fate of the bipotential gonad becomes fixed. So typically, we think of this as becoming a male or a female. If you have an X and a Y chromosome, you should develop testes. And if you have 2 X chromosomes, then you should develop ovaries. So when a baby's born, how do you know if it's a male or a female? The obvious answer is to look between its legs, right? But what if it's not so obvious and their genitals are ambiguous? What if their chromosomal sex doesn't match their genitals? So say, for example, they look like a female, but they have a Y chromosome. These are called disorders of sex development, or intersex, and they occur in about 1 in 5,000 live births. Intersex patients and their families face many challenges, both medically and psychologically, such as what sex should they raise their child as, and should they have corrective surgery or not? Our understanding of intersex is limited. About 20% of patients receive a genetic diagnosis, and this poses many issues for the management of patients that don't receive a genetic diagnosis, like could their gonads potentially become cancerous, so should they be removed? Or could they be left alone and preserve fertility? Or should we be looking out for other associated conditions, such as in bone growth or in the heart? One of the reasons that our understanding of intersex is so limited is that we don't fully understand the pathways of genes needed to make a testis or an ovary. So when we're looking for a genetic diagnosis, it's hard to even know where to start. My PhD is focused on understanding how we make a testis. So what we do know is that the Y chromosome contains a gene called SRY. SRY is needed to turn on the male fate. The main role of SRY is to activate a gene called SOX9. But what happens from here we don't fully understand. So SOX9 acts like a switch, and when it's present, it turns on genes required for the male fate, and I want to know what those genes are. So I have a mouse which is missing SOX9 in the testis, and using RNA sequencing, my lab has been able to identify 186 genes which were not turned on. So my question now is how some of those genes are contributing to make a testicle. To do this, I'm analysing gonads which are missing just that one target gene to see if they have any intersex features. Any new intersex genes I discover in mice will be added to the list of human genes which we currently screen patients for. Thank you.